Hey everyone, I'm Ross,
And I'm fitted.
and this is just a short clip of two humans talking about the NHS immunotherapy jab. So, this was on the news this week. The NHS just introduced this new jab for various different types of cancers, which is called a checkpoint blockade inhibitor. And we're going to go into how this works and what this means for patients who have cancer. So, I am somebody who works on immunotherapy. So for me, firstly, it's really exciting to see immunotherapy becoming more mainstream, becoming easier for people to access, and for something that's causing headlines and for the NHS to be embracing. I think it's all great news for immunotherapies. Immunotherapies, we talked about in a previous episode, are therapies that use the immune system to target and attack cancer. Your immune system will naturally try and defend you against cancer, but sometimes it doesn't work for whatever reason. And immune systems either help it to work better or reactivate it, or they can completely hijack it and reprogram the immune system more kind of direct in a more directed way. And this type of new jab by the NHS, it's an older, slightly more simple immunotherapy because it's based on an antibody injection. So basically, you may know what antibodies are. They're the sticky things in your blood that are produced by white blood cells, and they can stick to proteins. We call them antigens. And this immunotherapy uses an antibody that sticks to a protein on some cancer cells, make this protein. It will stick to that protein and block it from working.
Yeah. That sounds great. And this reminds us of me of the episode we did a couple weeks ago, where we went down into the weeds of how cancer evades the immune system and then how immunotherapy can target it. So in the specific case of checkpoint blockade inhibitors, what's happening and how is cancer removed?
Yeah. So checkpoint molecules are molecules that tell the immune system to stop working. So in your body, our immune system will fight off infections and it will become active and it will make you feel bad because when you have a cold, for example, that's your immune system working really hard to do things. It's not great to have it on all the time. And a checkpoint molecule or a checkpoint protein is what your body starts to make to tell your immune system to stop. We make them naturally in our body. Our body cells will start to produce them. But usually it's at the end of an infection. Cancer, some types of cancer will make the same molecules and proteins, the checkpoint molecules, because the cancer cells have learned that these shut the immune system off. So if your immune system is attacking the cancer cells, the cancer is always shuffling what proteins it's making, kind of mixing and matching everything that it has available. And when it finds something that gives it an advantage or helps it to survive, it will keep making that thing. If it's under attack by the immune system, the checkpoint molecules will help the cancer to survive and stop the immune system from killing the cancer. So the immune checkpoint inhibitors, what they do is that they block the checkpoint molecules from interacting with the immune cells. So they physically block them from binding to the immune cells and telling them to stop working. In other words, they free the immune system to work again after it's been shut down, unleashing it back against the cancer.
Okay, that makes perfect sense. And why is this revolutionary and how does it affect cancer patients?
So this is a type of therapy that's existed for a while because it's based on monoclonal antibodies. And we've been able to make these for quite a while because it's relatively simple to make antibodies in the lab. And we use them for all sorts of things. Like you can directly target certain things with antibodies or whatever. So the technology isn't necessarily new, but the revolution here is the ease of access. It's taken something that used to be a drip in a hospital that took hours. And now a patient can go and access this as a simple 1-minute injection because it's been formulated in such a way that we can now just give them an injection by a doctor and they go on their way. They get hours of their life back. It makes it so much more easy and pain-free for the patient. And there are thousands of patients who have this treatment to help in their fight against cancer. So this isn't something that all cancers will benefit from. You basically, what you do is you screen the cancer and you do a test on it to see if it's making that checkpoint molecule. And if it is, this is something that we can now easily give to people to help their bodies fight back against the cancer, as well as probably alongside other treatments.
So pretty much it means that we can administer this jab to a lot more people, right? Like we're not constrained by how many we produce. We were constrained by time mostly.
Exactly. It was wasting. If you have 14,000 patients on this drug and it takes a couple of hours for each patient. That's wasting 30,000 hours of the patient's time, the nurses and doctors' time, and we've gotten all that time back for everybody. So the drug remains the same that we had before, the same thing we were already giving people, but the time back and the convenience of the jab format is what we're gaining here. And it's bringing immunotherapy into the mainstream a little bit, which is cool to me. The NHS is embracing these therapies. They're becoming things that we can have easy access to. Because this therapy can sometimes be the only treatment people need or get for their cancer, but it can sometimes also be done in combination with other things. And that links us to the episode we did, which focused a bit more on CAR T cells, which is a much more complicated type of immunotherapy that I work on in my PhD at the moment. But these therapies are based on T cells. T cells are the cells that are being unleashed here. They're the ones that are being uninhibited by this drug. So if we could use this in combination with CAR T cells, it could be a way that we supercharge both drugs together.
How would that work? So can you please explain the synergy between CAR T and the inhibitors blockade?
Yeah. So the relationship between CAR T and the checkpoint inhibitors is CAR T cells are T cells, so they will also express PD-1. And I should have said at the beginning, by the way, that the way this works, the checkpoint that is being targeted is PD-1, and the cancer cells are expressing PD-L1, the ligand. PD-1 ligand is what the molecule is called. It binds to the PD-1 on the surface of the T cell. That signal tells the T cell to stop killing whatever cell it expresses the PD-1. It switches them off. And this antibody will bind to PD-L1 and block it from binding to PD-1 on the T cell surface. So the T cells are now not getting that switch off signal, and they're now free to go around killing again like they were trying to do in the first place. A CAR T cell is also a T cell. It's based on a T cell that's been modified in the lab. So they will also sometimes express PD-1. And the longer the CAR T cell is active for and the longer it's been trying to work for, the more PD-1 it starts to express on its surface. Because as I said, this is a natural reaction that happens. As the immune system is on for longer and longer, the body tries to switch it off to stop you having overstimulation of the immune system. So CAR T cells will get this just the same as regular T cells. So we can stop the cancer cells. Let me just add another bit. You also can't completely remove PD-1 from the CAR T cell. Like, it is possible to knock out genes and to remove proteins, but PD-1 has also maybe got some implications on how the CAR T cell works. So sometimes molecules that are to do with shutting off the system are also important in the signaling and the activation of the system. So you can't just remove it. But what this drug does is it blocks the cancer side. It blocks the ligand. So the PD-1 is still on the CAR T cell, still able to make the CAR T cell function properly, but it's not receiving that negative signal that tries to switch off the cell from the cancer anymore, which could supercharge the CAR T cells. There's still research being done. It's early stages with this. There's not that many studies that combine CAR T cells with other things yet, although they're beginning to do that, because they kind of want to see a clean product working on its own. But in the future, if CAR T cells already work pretty well, imagine how well they could work if we combine them with these other products that block the negative signals in the body.
Mhm.
That's why I personally find this quite exciting. So I think it could be another step forwards to CAR T cells and other immunotherapies becoming huge in the next 10 years or so.
Mhm. Sounds fascinating. This is the first step. And this is, as you said, is the first mainstream immunotherapy service or product that can be offered. But we have so many other things down the pipeline that will make this technology even more powerful than it is today.
Exactly. This is like the first step, but all the other stuff in the pipeline that's not been coming out yet, we're going to see it filtering into the system as this is already being done. And this is becoming a routine, easy therapy in the same way that I hope the CAR T cells and the other kinds of therapies that we work on in the lab right now will one day become routine and easy as well. So I'm excited for the future of cancer research and the kind of therapies that we're going to see in the next few years. I think it's going to be a huge amount coming out soon.
Fantastic. That's very promising, very interesting as well. I would suggest that if anybody has other questions about cancer or immunotherapy, to leave them in the comments, and then you can do another episode where we answer questions from the audience.
I would love to. Sounds great. Thanks for listening, everyone.